Great. Okay, now thank you all for having me. So my name is Gabe. I'm a fourth-year bioengineering PhD student down at Stanford. I'm working in Michael Fischbach's lab, and we generally work on the gut microbiome. And I gave this talk kind of generic boilerplate research title I usually give when I give more research-focused talks, but I guess Brandon mentioned that it might be good to talk a little bit about kind of just pathways into getting into research and how I got involved. So the way I'm going to try to split this up is do like a half and half. First half I'll talk about kind of my own path into research, and then second half I'll give an intro about what we do in the lab. I threw my email up there, so please feel free to shoot me a message after the talk if you have any questions. I need to take off right after this, but I'm usually pretty responsive on there, so please feel free. And I'll throw that up at the end too. And I don't know if this is a kind of required talk or not, but if it is, some of you might be zoning out for the next hour, so I'm going to give you kind of key takeaways of what I think you should be learning during this talk, right? The big one I want to hammer home is just kind of growth, right? So I remember when I was in college, which wasn't that long ago, but maybe seems like it was long ago for people sitting in the audience. It felt like that was it, right? Like I was making decisions, or like that was the end of the road, like what I decided today was going to determine what I did for the next like 20 years, and that definitely was not the case, right? When I was in college, I was doing things that were completely different than what I ended up doing right now. And I kept just trying to learn new things, keep seeing what's out there, and that has paid off big, I think. I'd be a big advocate of just learning as much as you can, just getting new experiences over and over again. Because you can end up getting paid to do research, which is what I do now, and it's awesome. Really, and I can't kind of emphasize that enough. And I think that's an option that a lot more people should consider when they do. Like I just said, staying connected to science and technology is super rewarding. Having just made a pitch for getting paid to do research, I think just staying connected, no matter what, is really cool. Whether that's in your day-to-day work, you're doing bench work, or you're doing kind of communication, or you're just kind of reading on your own time. I think it's super important to stay connected after college. And that doesn't always happen. So I'd say that's important. The last thing is, if you don't know what you want to do, and I definitely did not know what I wanted to do in college. I can't tell you 100% I know what I want to do right now. What you can always do is focus on skill sets, right? And focus on just learning things that are going to be useful no matter what you do. In my case, that was kind of programming and math. And like I'll try to tell you guys, that opened a lot of doors for me. And I think that specifically that's a skill set that will open a lot of doors because it's so general and it's so in demand. And really the barrier to entry is kind of low. You can pick it up on your own, you can do a lot of self-learning, and I'd advocate for that really strongly. And also, I think it's a small enough crowd where we can be pretty informal, so if there are any questions, feel free to interrupt, like doing these or just shout something out, you don't have to raise your hand. With that in mind, let me jump into kind of personal intro side. And then here's my paper version of me, which is mostly just my academic history, but I also want to mention, I was talking to Rance before, I grew up in San Francisco, over in the Sunset, near kind of UCSF Parnassus. We were talking about how the city has changed, and it definitely has. I felt that, for sure, but still proud to have grown up there. I'm first generation US, my parents are from Brazil, so I actually grew up speaking Portuguese at home, and really did not speak English super well until I got to school. I remember I kind of locked myself away over the summer. I used to watch kind of just the crappiest sitcom, action movies I could find just to pick up English, right? I don't know if that makes sense, but that was a big deal for me right now, right? In college I ended up doing computer science and then ended up doing a master's in applied math, which led into this PhD in bioengineering I'm doing right now. And that's just to get back to that point that when I was in college, I really had no idea what I was going to do. And I was not a good student. I was pulling pretty bad grades on paper early on. The one thing I knew was that I kind of liked coding day to day. I wouldn't even finish my assignments. I was getting really bad grades. But I at least liked the day-to-day version of it where I'd go home at night and just kind of mess around with code. And I kept doing that and kept doing that, even though my grades were bad, I just kept getting better at it. And that paid off in the end, even though the first couple of years were super discouraging. And I think in college is something that you don't really realize early on, that you got to keep growing that skill set, even if you're not getting the paper grades early on. The other thing I want to mention is that over this trajectory, I kind of started getting interested in biology. I think biology is awesome. I think it's a really cool time to be doing biology, and I knew nothing about it in college. And the way I ended up kind of getting exposed to that wasn't necessarily through coursework, because a lot of the classes were full of people who knew they were doing biology early on. It was through kind of summer experiences. And I'll mention one of those that was super important to me. But when I didn't know what I wanted to do, I just kind of reached out to people and asked them if they want to take an intern on, if they had openings for summer positions. And like I'll mention in a few slides, I did that a lot, right? Didn't really know what I was asking for, but I just asked everyone if they were just willing to take on, an undergraduate, just looking for experiences. Eventually, that kind of led to things that really showed me what was out there and gave me some skill sets that really ended up helping down the line. So I'd really recommend doing that. Just look for as many different experiences as possible in just a variety of fields if you don't know what you want to do. I'll talk about kind of what I'm doing later on too. Very broadly, I'll work on kind of computational methods and gut microbiome research right now. Going back to kind of college, when I didn't know what I wanted to do, I actually was up in SF for a summer and I went to Green Apple Books. I don't know if any of you have been up there, but it's a cool used bookstore. And I found the book over on the right called Regenesis, and it was like on sale, like $5 or something. And I just picked it up kind of on a whim and read it. It's by this guy, George Church, who I didn't know at the time, but he's this really famous kind of geneticist. And if you Google him after this, or right now, I've got to laptop out, this guy is into some really, really crazy stuff, right? He's kind of kooky. He wrote this book that's this like manifesto on how biology is changing the world, right? And genetic engineering is this like amazing thing. It's gonna like change everything about everything. It sounded very like startup-y, right? But I started digging into this and saw that it was really cool. So this guy had some pretty crazy projects going. He still has them. One of them, he wants to bring back woolly mammoths. He dug up some woolly mammoth DNA and he's working on this project to clone woolly mammoths. He wants to make like an army of them. The other thing is that he's working on DNA storage. So storing like digital data in like strands of DNA. And I had no idea what the application for that was, but I thought it was awesome. Right? So these guys they took a gift of like a running horse and then they just like encoded it in DNA and bam. Which is storing things in DNA. I really had no idea what the practical purpose of any of that would be, but I just thought it sounded really cool. Right? It was a great dinner conversation. So I kept kind of sniffing around at that, and I said, okay, I don't know anything about this. I know how to code a little bit, but I know this is awesome, and I want to get involved. Right? So what I did, I had a summer coming up. It was between my junior year and my senior year. And I said I wanted to spend that summer just kind of learning more about biology and getting experiences that would open doors for me. Right? But I had no idea how to get involved in any of that, right? How to get involved in the professional research world. It was totally kind of dark territory for me. So what I did is I wrote this form letter, right? Which is, people tell you this is like exactly what you're not supposed to do when you like cold call people, right? They say you're supposed to write some kind of email that's like very personal, that you like research this person very well. I did the opposite. I wrote the most generic email you could possibly write. I dug this out the other day when I was putting these slides together. You see it from 2013, right? And I just said like, hey, my name is Gabriel. I'm majoring in computer science and minoring in neuroscience, and I spiced it up saying I was doing quantitative computational neuroscience, but it was really just neuroscience. And I said I wanted to get more involved in whatever research, right? And I literally would just copy paste whatever was on the lab website, and I said like, I want to get involved in this research. Always have wanted to do it. Love what you're doing, so interested. I'll do anything. Like, just please take me over the summer, right? And I emailed a bunch of professors from the Bay Area, and I said, look, I'll eat more for free. I'll get coffee if you want, right? As you can imagine, mixed results, right? So when I was digging through my Gmail inbox, I found this week where I had like sent out this exact form letter, and it's kind of grainy, right? But I don't know if you can see this. This is from like April 2nd. This is from a single day. And this is actually 3 pages, all with the exact same subject, right? The exact same form email, and I sent it to hundreds of professors. That's not even an exaggeration. As you can see, like, very few people responded, right? The hit rate was super, super low. Almost all of these are pretty low in email threads, right? A couple of these, there's like one reply. And if I was lucky, these professors would respond to me with like a single sentence saying like, no, sorry, good luck. over here, this is where the breakthrough happened, right? And this was, one thread. There were literally hundreds of these emails. And finally, this one kind of random postdoc responded to me from this neuroscience lab at Berkeley who said, all right, fine. We'll take you. I need someone, send me your resume. So I can make a resume and I sent them the resume. And so she took me on. And I didn't know anything about that lab. I didn't know anything about what she was doing. I just wanted to try her research, right? What ended up happening was that I worked on something awesome. And I had no idea what any of this was beforehand. I just knew how to code, and I knew I was interested in neuroscience at the time. But it turns out that the project I was working on was kind of this product of these neurotechnologies that had been coming out, the years before I was trying to get a research gig. And it just so happened that because these technologies had been developed, there was research being performed that required some coding, right? So let me backtrack for a second and just introduce what these technologies were. Over on the right, right around 2011, early 2010s, neuroscience researchers started realizing that rats and mice have empathy. Right? And when I say that, it doesn't sound like mind-blowing, but in the neuroscience community, that was like a huge deal, right? So they came up with this experiment where if you take a mouse and if you trap it in the, I think I have a point here. If you trap a mouse, right, in this little plastic cage, there's actually a lever up here that the other mouse can pull to free its buddy. What you'll see is that the free mouse will free his friend, right? And if you actually lower a microphone really close to the cage, you can actually hear the trapped mouse kind of screaming for help. It's terrifying, right? But neuroscience researchers were blown away that mice would help each other out, right? So they started doing these experiments more and more. If you actually, they have a pretty twisted experiment where if you do this, you have the trapped mouse over here, and then you pull a pile of chocolate over here. Actually, the mouse is going to help out his buddy before going for the chocolate. He's going to save some chocolate for the cage mouse. So that's a big deal, and that actually still is a big deal in neuroscience because we're saying that empathy is kind of this universal thing, pro-social behavior. It's not just humans who care about each other, right? At the same time, a little bit earlier, Boyd and Zhang in this lab up at Stanford actually developed this technology called optogenetics. Some of you may have heard of this. This is a really popular technique, increasingly so, where there are certain light-sensitive viruses, right? And you can actually infect rat brains with this light-sensitive virus in certain areas of their brain, and you can put an LED stimulator, so that's what that little cord coming up the top of their head is. If you turn on the LED stimulator, you can selectively turn on, certain parts of their brain, which is really cool because you can choose which parts of the brain you're turning on. You can kind of turn on behavior at will. You can see what happens. So I got in touch with this postdoc who wanted to turn on certain parts of the brain and see if she could kind of toggle empathetic behavior and see what parts of the brain are involved in empathy and pro-social behavior. That ended up being important because that meant she needed a programmer to make a kind of computer vision software package for her, right? So what we're looking at here is actually from the experiment we were running. This is the arena she had set up. So this is actually a live feed of, we actually transitioned to rats. So here's a rat trapped in a cage, and then here's a free rat, and we're measuring how often that free rat is going to go and help its buddy out in the cage. And let me see if I can get this video working. So what we needed was something that took a live kind of webcam feed, and this was actually a webcam, right? We needed to track where these two rats were, and that would feed back, keep track of where they were, turn on the optogenetic, the LED stimulator in real time, based on the way this rat was moving, right? So this was all written in code that I was writing in Python. I don't know if any of you are Python programmers or tried it out, right? This was all in Python, and it was using like a $5 webcam. And it was just keeping track of this rat, and if it was straying too far, hanging out over at the corners of the cage, we would turn on the LED simulator and see if we could encourage it to help out its friend. And that was awesome. I mean, that was so much fun. And I realized that, something I was worried about, which is that, you know, if I kept doing programming, I was going to have a tech company, you know, working on a loading screen for a social network or something. And if you want to do that, that's great. I didn't want to do that. I knew I didn't want to do that. And I found something that was way cooler, and I realized, like, this thing that I like doing day to day, right, which is programming, I love doing that, could be married with, you know, a cause that I believe in a lot more than the other options, which was just biology and biological research, right? Just kind of understanding how biology works. At the time, that's how the brain works, but even that changed over time. And I think the takeaway I just want to drive home here is I had no idea any of this existed, right? Even when I was sending out those hundreds of form emails. You know, this was one lab in a sea of just random lab pages that I landed on. This one was at Berkeley because it was local. It was all these kind of coincidental things. And it ended up being really cool, and it helped me realize that what I want to do is work on software, you know, and computational tools to move forward biological research. And that all happened outside the classroom. And so I can't emphasize that enough, right? Like, I think most of the meaningful things that happened to me were experiences outside the classroom where you realize what's really going on, what's cutting edge, what are people doing out there. And I think that's super important, especially in research, when the live version is pretty different than the textbook version. If there's no questions on zapping rats, I'm going to maybe give a brief overview of my own research now, which is pretty different from what I was doing before. And this is something I didn't really know anything about until I got to grad school. Just another version of trying to keep seeing what's out there. Let me give the takeaways for this part, right? And then you can go back to sleep. The gut microbiome, maybe some of you have heard about this. This is a big one, right? This is a huge deal. This is kind of a cutting edge topic in science today, and I'll try to talk a little bit about what I do specifically in that. There are big implications for a bunch of important diseases that people are really interested in. Two of the big ones right now are autism and metabolic disease, but there are implications, and I say those two very carefully because the links are not well studied yet, right? But there are a whole host of kind of health outcomes that people think are related in some way to the gut microbiome. If any of you have heard about the gut microbiome, you've probably heard about kind of probiotics, right? And so I think that sort of thing encourages you to think that when you talk about gut bacteria, it's as simple as saying, like, okay, there's good bacteria and there's bad bacteria, and I just want to, like, crank up the good bacteria and, like, get rid of the bad bacteria. It's not like that, right? And I think that people who will try to sell you on that idea are usually trying to sell you on something else right after, right? And that those probiotics are based on that. And one key takeaway is there's more going on underneath the hood, and that's a very active area of research right now. The active area that I focus on is the actual metabolites that are involved, right? So these are the small molecules that you're exchanging with the gut bacteria. And I think I have a picture down the line that kind of illustrates that a little bit more in a second. The final point, and this is kind of a big takeaway, is that in order to do all that, to figure out what's going on in the gut microbiome, to push research forward, we need computational tools, right? And the big takeaway from this kind of slice of research, but research in general, is that science as a whole is getting way more computational, right? I'm sure all of you have heard of, like, big data, right? All these buzzwords. That's happening in science too, right? Scientists right now are pumping out a lot of data, and the people who are pushing things forward a lot these days are people who are able to kind of harness all that and be able to say things using computational tools. Okay, let me start out with kind of a scary thought, which is that if you were to take a sample of your own blood, right, look like that, you could let it sit out for about 30 minutes, and after about 30 minutes, it would separate out into kind of the heavier portion, and then the yellow portion on top, right? You've probably seen something like this. You take away what's at the bottom, right? What's at the bottom are your white blood cells, your red blood cells. This is kind of the stereotypical what's in your blood, right? That's your immune system kind of functioning, and that's also your oxygen carrying kind of units, right? That's going to leave about half of your blood by volume sitting up there in the yellow part, right? That's your blood serum. And one of the crazy things is that your own blood, if you were to give that to a doctor or a researcher, hand them that yellow portion, they actually can't tell you what's in there, right? If you ask anyone right now, what is pumping in your veins, they can't give you an answer. We don't know. We know some things, right? And the things that we do know are definitely in there are a huge deal, right? These are things you've heard of because they're pretty important. Probably heard of cholesterol, right? You probably heard that cholesterol can be bad for you, et cetera, et cetera. It's really important because it's a quintessential kind of part of your body's functioning, especially via cell membranes, right? Steroid hormones, you have things that are controlling your immune system and your metabolism flowing around in there. You have neurotransmitters, so those are things that are regulating kind of brain activity. And then you have things that control things like pain inflammation, right? These are all huge kind of physiological functions of your body, and the reason why these things are floating around in your blood is because once in your blood, you can access, every single organ system, right? So whatever is in your blood is pretty important to figure out, and you should all be terrified right now. So I can see you all are very terrified that we don't know everything that's in your blood, right? Let me tell you about one molecule that we recently figured out is in your blood, and we're sort of figuring out bit by bit what this molecule does. Right? So this molecule is called indoxyl sulfate. I don't know if anyone in the room is kind of a chemist or an aspiring chemist, but it doesn't look like a crazy thing, right? But what we're seeing, and so I'm actually showing you some unpublished data, so no one take pictures of this, please. But this is from a lab we collaborate with over at the Cleveland Clinic. Okay, well, we'll block it out. It's all right. That's fine. This is from the Cleveland Clinic, right? This is an ongoing study they have of 4,000 patients, and they're tracking a couple of different molecules in the bloodstream. What we're looking at over here is the amount of indoxyl sulfate floating around in the blood divided up into quartiles, right? So Q4 is going to be the patients that have the highest levels of indoxyl sulfate. What's going on the x-axis over here, we're looking at the hazard ratio, right? So this is the probability that you get something called MACE, right? It's a major adverse cardiac event. Not good stuff, right? There's a heart attack, stroke, cardiovascular death, not things you want. And when you look at the graph, you can see that as you go down to Q4, your chances of having one of these heart attacks, strokes, cardiovascular death increases almost twofold, right? And that tracks very neatly with this molecule indoxyl sulfate. Okay, so we say there's probably other molecules in the blood doing crazy stuff. We know this one's probably bad, but so what? What I want to point out now is that this molecule indoxyl sulfate, the other half of the story we're figuring out now, is that this thing comes from gut bacteria, right? And this is a great microbiome story. This is what people are super interested in, right? You eat dietary tryptophan. We're all eating tryptophan all the time, and we eat tryptophan. And something like 98% of us right now in this room have this one strain of bacteria called Bacteroides beta-omicron. You can only see it down there. There's one bacteria, it has a gene called BT1492. And that BT1492 is going to take that dietary tryptophan, that's in almost everything you eat, and it's going to push it down this chemical pathway, hand it off to your own endogenous enzymes, and at the end of that chemical pathway, you get this molecule indoxyl sulfate. So if you took that bacteria out of you, you wouldn't have indoxyl sulfate floating around in your bloodstream, right? Turns out that this molecule, along with some other ones, are part of the reason why things like red meat, some food you eat, can actually cause cardiovascular death, right? You think about foods that cause heart attack and stroke and whatnot. This is the sort of relationship that you've got microbiome in place with the food you eat to produce molecules that end up being bad for you. So let me give you a picture of what's going on in the gut microbiome and tell you that as far as we know, and this is a changing number, more than 10% of the molecules that are floating around in your bloodstream right now come through the gut bacteria. Right, they're dependent on the presence or absence of certain gut bugs, and that depending on what you eat and what you feed them, you can actually control what bugs are there and what bugs aren't. Right? So right now we're looking at a cross section here. We're looking at a very simplified version of what the inside of your gut looks like. Right? So on this side of the gut, this is your gut wall. On this side of the gut, you got your endogenous physiology. This is your organ systems. Right, we'll live here, but this is your bloodstream. Over up top, we're looking at the inside of your gut. Right? which actually doesn't access any of your native physiology. Right? The way my boss talks about this a lot is that humans are just hollow tubes, right? We're just organs wrapped around like a hollow tube, and that hollow tube starts at your mouth and goes all the way down at the other end, which is a really comforting way to have your boss talk about humans. This is a good picture of what goes on in the gut microbiome. Right? You do your thing. You eat whatever you choose to eat. Bacteria, fibers, prebiotics, et cetera, right? The gut bacteria that are living on the inside of your gut do their thing on their side of the gut, right? They will chew up whatever you eat, they'll ferment that, and then they'll spit out a bunch of molecules. Those small molecule metabolites, that's what seeps into the inside of your gut. That's what gets into your bloodstream, and that's what has the health effects that are associated with the gut microbiome, right? There are different strains of bacteria that are present in different people, and if you feed them different things, they'll do different things to you, right? So you've probably all heard that fiber is really good for you, right? You should eat a lot of fiber. If you talk to microbiome researchers, they eat a disgusting amount of fiber. So I was listening to a story the other day. There's a microbiome conference that my boss went to, and apparently at the cafeteria where they were hosting it, the salad bar ran out in the first 20 minutes of the lunch break, right? And then the grill was like totally empty. There was no line. Everyone was eating a bunch of fiber. The reason you want that is because there are a very common set of bacteria that hang out on the inside of your gut. Almost all of us, since we live in America here, have this kind of bacteria. They'll chew up dietary fibers, right? And then they'll spit out these short-chain fatty acids that do a lot of good stuff to you, right? They make you less fat, and they make you less likely to have diabetes. And so that's another big takeaway. Eat a lot of fiber. And if you're not eating salad, take Metamucil, psyllium husk. Apparently that works. Alternatively, if you're eating a lot of salt, a lot of sodium, you can inhibit a lot of that good stuff I just mentioned, right? The takeaway is that, we think about food as being good or bad for you, but we oftentimes don't think about what's going on underneath the hood. We just kind of take it for granted, right? What's really going on is that you have a bunch of these little armies of reactors that are hanging out, chewing up the food you eat and doing good or bad things with it, right? So wouldn't it be cool if we could engineer gut bacteria that would turn burgers into good molecules for us? Sure, that's the dream. Down the line, not what I do, unfortunately, but maybe one day. If you're interested, I'd highly recommend a career in microbiome research. Let me tell you right now about what I do, which is way more boring than what I just said. So I just mentioned that we don't know what's in your blood, right? We don't know all of what's in there. We definitely don't know everything that your microbiome makes. Even though we know a lot of the strains, we have a hard time figuring out what molecules they make. Why? The reason for that is that the technology you would use to try to figure out the molecular composition of a liquid mixture is typically mass spec, right? So liquid chromatography and mass spectrometry. I don't know if any of you have had a chance to kind of check this out, maybe read about this or run one here. This is technology that's been around for a while. The problem is that the data that comes out of it is super messy. Right? So actually getting the samples, which is your urine sample and blood sample, as you can tell over there, is the easy part. Right? It's easy to get a urine sample from someone once they agree to do it. Running it through the machine, not so bad. Right? What happens is that you get data that looks super messy. Right? So what the LC-MS is going to do, two parts, liquid chromatography as the first part, the LC, MS, mass spectrometry, is the second part. The liquid chromatography is actually this stack over on the right side, and that's going to separate out your liquid mixture into its individual kind of chemical components, right? So you actually drag the liquid mixture across this chromatography column. The molecules are going to pop out one by one, and then you're going to actually fly them across this tube, at least in this type of mass spec, and you're going to weigh them, right? But you're going to weigh them super precisely, so that weight should tell you a lot about their chemical structure. This is what the raw data looks like, and this is actually pretty faithful, even though I stole it from Wikipedia. This is pretty faithful to what it looks like when it comes off the machine, right? You're measuring your data in 3 dimensions. On one dimension, you have your retention time. So this is the time that your molecule came off the column. You got your M over Z, this is your weight. That's a function of the chemical structure of the molecule you're looking at. And then you got your intensity. which tells you how much of that molecule is in your sample in the first place. And you just take that and you say, okay, here are the molecules that are in there. It's kind of bad, right? You have no idea where one molecule stops or one molecule ends. That's kind of a challenge there, right? So what people will typically do is they'll generate this kind of data, and then they have like 5 molecules they know that they're looking for beforehand, like cholesterol or some steroid or whatever. And they'll say, okay, I know that cholesterol weighs, this much, and I know it should come off my column, it's like 30 seconds in, right? So you go over to this part of the data, and you'll say, yeah, there's a fat, there's a big piece of signal right there. That's cholesterol, we saw cholesterol, we're done. Let's get out of here, right? What we're trying to do in the Fischbach lab is trying to expand our capabilities, so we're looking at the entire data and saying, what's in there? What are all the molecules that are in there? How can we expand our identification capabilities? Let me also introduce the idea of how we would kind of tease out which molecules are specific to the microbiome, right? So I just told you that you have all these bugs living in your gut. If you don't want bugs living in your gut, if you're a mouse, one thing you can do is you can grow up in sterile conditions. So there are actually labs that have these, they look like little space stations for mice. You just raise them in completely sterile conditions, right? They're bubble blows, but they're mice. And if you're super careful, there's no bacteria anywhere around them for their whole lives, and they're completely sterile. They have no gut microbiome. And then if someone punches a hole in that, all the bacteria get in, and then years worth of science is ruined. That just happened in our lab a couple of months ago. But if everything's working right, you have germ-free mice, right? You can compare them to mice that are raised in normal conditions. They have normal microbiomes, and you can do what I just described. You can do LCMS and look at the signals that come out. And you can say, okay, here's a molecular signal that looks like it's really showing up in my conventional mice, and then here's a molecular signal that looks like it's totally absent in germ-free mice. The germ-free mice are fine. They are kind of weird. They get sick really easily, and a lot of other things, and that's how we figured out that the gut microbiome can be really helpful for them. But they do stay alive, and you can compare their molecular signals, and then if you're really lucky, you can identify what that molecule looks like once you've identified a cool pattern separation there. And that's how they figured out that that molecule, oxyl sulfate, is a gut microbiome molecule. What I'm doing now is a project working together with Tim Meyer, who's a nephrologist. He's a doctor at the Stanford VA hospital. Jakob Brynjak, who's more of a chemist type than I am. I'm kind of the programmer guy, and he's the chemist guy. What we did with Dr. Meyer was we got samples from people who have undergone total colectomies, right? So you cannot raise people in bubble boy conditions, unfortunately. But what you can do is look at people who have had their large intestines removed. And this is actually a really common medical operation in the US for a number of reasons. If you've heard of irritable bowel disease or colon cancer, those are 2 reasons why people very frequently will get their large intestines removed. This is interesting from our point of view because even though that wasn't the purpose of the operation, when you took away their large intestine, you ended up removing most of their gut microbiome, right? So now we can do what we would do in the germ-free mice, except in humans, which is way cooler because there's a lot more going on in humans than in mice. And we can say, what's the molecular signature of our healthy patients versus our collecting patients, right? What I have been working on recently has been going back to kind of math and coding and taking a look at these molecular features and figuring out how to come up with a distance metric, right? So a lot of you are probably taking some kind of linear algebra or some kind of math, right? What I'm doing these days is all about kind of defining a mathematical function between 2 kind of featurized chemical signatures to say, how close are these chemicals to each other? I want to measure, literally measure from -1 to 1. Like given 2 of these kind of molecular input signals, spit that in there, right? And they call me either super similar, or these are not similar at all. And that has allowed us to develop kind of new computational techniques for getting at that huge grid of molecular signatures and teasing out what's in there, where the molecules that we've never kind of figured out before. If any of you are taking any kind of programming that involves clustering, right, like data science techniques, like totally recommend you read up about that if you're interested. I want to show something we saw early on, which is the raw data that we took from that LCMS run. So what you can do is draw a bunch of boxes around those raw data peaks, say here's one molecule, here's another molecule. Each one of these is one of those molecules we found in the raw data. And using that technique I just mentioned, we defined a distance metric. And so we found 55 peaks that are all very similar to each other in that kind of chemical space, right? And now we're looking at their intensity profiles between our collecting patients, no large intestine, and our control patients, these guys have large intestine. And what we're seeing is what I showed you in that kind of cartoon version of the mouse, right? This is an actual kind of raw data signal where we're seeing very high intensity in our control patients and kind of an obliterated signal in our collecting patients, right? And it turns out that this cluster corresponds to indoxyl sulfate and a bunch of other kind of related molecules, which is very cool. We're seeing this in real life, we're seeing this in action, we're seeing this in people, in mice. So that's been a big deal. Let me introduce MS2 very quickly, just to tell you what I'm doing with that. And fair warning, it's kind of boring. But the cool part of this is that we borrowed techniques that people are using to mine text, to mine kind of chemical structures. And I think that's really cool. I think that techniques people are developing in data science, and text mining especially, are worth reading about because they can apply to anything. Something as random as figuring out chemical structures in microbiome data, right? But before all that, to get more detail about a single molecule of interest, right? You've isolated a peak, you can do what's called tandem mass spec, right? You can say something that weighs exactly this much of the molecule I'm interested in. I'm going to run it through this collision cell, and this collision cell is full of inert gas, right? So what you're actually doing is you're just blowing up this molecule. You're doing this at super high speed, and you're colliding it with a bunch of inert gas fragments, and that molecule is going to fall apart in that collision cell. And everything, all those fragments are going to fall out the other end, and you can measure the mass of each one of those product fragments. What you end up getting is raw data that looks like this, right? So what we're looking at here on the x-axis, this M over Z, this is your weight. And then on the y-axis, we're looking at intensity. So how much of this was in your molecule? We're looking up here, this is the weight of your entire molecule. And each one of these little peaks is an event where the molecule broke apart. Well, a fragment came out, we measured that weight. You know? That fragment broke up, ran into some more gas molecules, we measured those fragments. You measured the popped out weight, and that keeps happening over and over again. We're just chopping up this molecule over and over, looking at the weights, right? And then our job is to figure out, just looking at these little sticks, right, what the chemical composition might be, what's the structure there. That's an open question right now in metabolomics. What I have been doing in the lab is borrowing this technique from text classes. And if nothing else, if you're not into chemistry, if you're not in microbiome, at least check out Layton Gear's application, because this is really cool stuff. What you can do, this is a model, a statistical model, that lets you take a bunch of text, right? It lets you take a text corpus, and it'll let you fit them all, right? You make some assumptions. So this is now in some statistics, but you make some assumptions as to what the kind of underlying generative process might be. You make those assumptions, and what comes out of your text corpus is a bunch of topics and a bunch of words, right? Your topics, you have a finite number of topics. Each one of those topics has high probability words. So what that would look like in practice is you run this model on a bunch of text. You can look at a single text, and you can actually say, what are the high probability topics that came out of there? I don't know what they were beforehand, but the model fit this, right? This document, for example, has like 2 high probability topics, yellow topic and blue topic. Who cares? But except that I can actually go in and look at the high probability words in those topics, right? So I can take a look at the yellow topic and say, this topic, high probability words are election, senator, lobby, official leader, right? Which probably has to do with politics. Our blue topic looks like it has to do with baseball, right? Got ball, bat, player, etc. The way the model's set up is you can actually have words that overlap between topics, which is why it's been pretty powerful in the past. So if you have like Washington, D.C., maybe that shows up in both of your topics, right? Really flexible, and people have been doing this on things like Wikipedia, right? So you can take all of Wikipedia, you can train a model, you can train an LDA model on there, and see what pops out. You can see Wikipedia actually broken up into like a bunch of different topics, right? And then you can take a new document and say, like, without reading it, which is great, if you want to be late, you don't want to read the document, just run it through the model and say, I know the 5 topics that this document is about, right? Is it worth reading or not? Oh, I'll take a look at the topics. What I have been doing recently has been translating this type of model to work on chemical data, right? So everything I just described was in the text world, right? And these models were developed with text documents in mind. But what we realized in the lab is that translating a kind of chemical data into something that looks like text can let us apply this breed of model to this type of data. And now we're just kind of switching words around, right? We're not looking at topics that are made up of words anymore. We're looking at topics that are made up of molecules, right? So now our topics, instead of being politics and baseball, look like chemical substructures. So the idea is that we take one of those MS2 spectra that I showed you before, which are hard to pick out. We can train a model, run it on one of these spectra, and what pops out is something that looks like this. You get 2 high probability topics, and these topics tell me, you know what, in this spectrum, it's pretty hard to interpret. We probably have these chemical substructures, and that goes a long way in helping us identify what the molecular signatures are in our raw data. Super cool. So this is all done computationally. And then what I did here is I just threw up a bunch of raw data from the lab. But the point is here that we can move quickly. So over on the left, this is all publicly available data, right? You can go on your laptop right now. You can go to a database. You can get free MS2 spectra. I took 1,600 of them, and I trained one of these LDA models. None of our in-house data, right? So this is all from the trained model. This is one of the 200 topics of the model. This is topic 144, right? And the way that we're dealing with the raw data, I can take a look at the topic and take a look at what my high probability words are, whatever. These are all molecular formulas, but if you're around these long enough, you take a look at that, and what pops out, you say, that's definitely a steroid core, right? That looks like that chemical structure. And by the way, if I go into those public MS2 spectra that I trained the model on, and I look at the high probability documents there for this topic, most of them are steroids, with the exception of these ones that are labeled in there. What's even cooler is if we go into our data that we've been painstakingly pumping out, this is data that takes a long time to pump out, and then to label, I can run it through this trained model in about 2 minutes, right? And I can get probability rankings for the probability that all of these MS2 spectra have a steroid structure in them, right? So I can label the data we've been churning out as yes steroid or no steroid. And what's really cool is if I look, I had a bunch of things that we had figured out were steroids on our own, and this took us 6 months to do, and this came out in 2 minutes. What's even cooler is that there were 3 spectra in here that we could even get to. We couldn't figure out what they were. This model figured it out in a couple of minutes, right? And said, hey, you got more steroids here, you missed those. And by the way, if I take a look at the raw data, now going back to our collecting patients versus our control patients, this is worth knowing because these are high intensity in our control patients, and they're obliterated in collecting patients. You got to figure out what these molecules are, right? These are new things that you weren't able to figure out. So that's been great, because that's allowed us to assign kind of molecular identities to things that people have not done before. We can do it faster, and we can do it more accurately than people have done in the past. And so that is what's going on right now in the Fischbach lab. In summary, there's a lot to learn still in the microbiome field. We need to figure out what these molecules are, and that is the tip of the iceberg, because once you figure out the molecules, you got to figure out what bacteria are involved, you got to figure out what genes are involved, you got to figure out what you're eating that's going to activate these genes, that's going to produce the molecules. But the first thing, you've got to figure out what molecules are present. This is stage 1, and there's plenty of time, plenty of work to still do in the microbiome field. The thing I want to drive home is that, at least in my experience, techniques in computer science and math have really gone a long way. What I was just showing you was one example of a technique developed using kind of data science, computer science approaches that are beating people who are trained as chemists, right? And that's happening across the board. Microbiome research, biology, science in general is getting more computational. I don't think that needs to be a bad thing. I just think it means that people who want to do scientific research have to be conversant in computational techniques. And that's my kind of driveway. You don't need to be a biologist. There are pros and cons there. And please feel free to shoot me an email. I'll put up in my slides. if you want to hear more about what that is like. But no matter what you do, a lot of people these days are applying these kinds of techniques across the board. And knowing how they work and how to program and the underlying math can really help you out across the board. And in my case, I didn't know what I wanted to do, so I kept learning that kind of stuff, and it opened a lot of doors for me, and I'd recommend that if you don't know what you're trying to do, do that, right? Or at least keep doing things that you like doing, and try to learn as many skills as possible, and keep an open mind. With that, let me just throw up some acknowledgements. I've collaborated with a lot of people on this project. Special thanks to Michael Fischbach, he's my PI, and then Jakob, the postdoc I work with, as well as Tim Meyer, a clinical collaborator. I also received funding from the NIH Biotech Training Program, so thank you to them for that, and my collaborators across a couple of labs. There's my email again. Please feel free to shoot me any questions you might have. And any questions now I'd be happy to take as well. Otherwise, thank you for having me.
Michael Schneider's lab with the iPhone stuff?
The big data? No, but that is a really cool project. And they're collecting, the scale of data they're collecting is orders of magnitude bigger than what we can do in our lab. And I think that they're still collecting, but they're going to come up with some really cool things. A big difference is that they're doing higher level data, right? They're collecting kind of physiological heart rate, and things that you can measure from outside the body, and then we're doing a lot more kind of molecular level things you can measure from inside the body. But very cool. Michael Snyder at Stanford Bioengineering, really great stuff.
I wanted to read these.
Is that right? Very cool. Thank you for your service. Great. Okay. Thank you all for coming.
Or something like that.
That's what you